Hi everyone, my name is Nadia Fernandez, and I'm a first-year master's student at Purdue University in Andrew DeWoody's lab. And today I'm going to be talking to you about some work I participated in as an undergrad that has been recently published in the Biological Journal of the Linnean Society. Okay, so to start off, in conservation genetic studies, effective population sizes can be used to understand genetic drift and allow us to predict the loss of neutral genetic diversity. For example, looking at this figure, we can see that larger effective population size can help maintain high levels of genetic diversity over generations or time, but lower effective population sizes can result in the loss of genetic diversity over time. So, and this ultimately can lead to declines in diversity that can reduce your evolutionary fitness and lead to smaller population sizes. Okay. However, to combat these losses, gene flow or migration can help facilitate allelic exchange, which can increase your effective population size and thus increase your genetic diversity. Migratory species require at least two distinct habitats, and in turn these can result in these species being more likely to be impacted by ecological changes such as hurricanes, such as competition, predation, or pathogens, compared to species that only require one habitat. However, other factors such as biomes may be influencing genetic diversity. Therefore, one of the objectives of this study was to test if migratory vertebrates harbor less genetic diversity than non-migratory species. And we hypothesized that migratory vertebrate species are more likely to be impacted by fragmentation and suffer population declines, which can result in the loss of genetic diversity. In order to test our hypotheses, we utilized a number of published microsatellite studies on wild vertebrates where heterozygosity and allelic richness estimates were obtained. In brief, I can give you, I'm going to talk about some of the criteria we used. We utilized studies from 1990 to 2014, studies that had at least 10 individuals, at least 5 loci, and also in order to and we utilized studies that excluded threatened species to avoid any potential effects of migratory behavior and conservation status. And on the diagram below, you can see what our distribution was among our taxa after all these filtrations. Next, we scored migratory behavior of each species, and for the purposes of this study, we defined migration as a predictable mass movement of individuals that occurs in the same direction or towards a particular common habitat and is a further distance traveled than typical daily movement. And in order to define these movements, various databases were used for taxa to provide formal description of each species. So to test the effect of migratory behavior on genetic diversity, we utilized a linear model, and we bootstrapped the linear model coefficient estimates to assess significance. And our results for the interaction, here is one of our results for the interaction between class and migratory behavior, predicting genetic diversity. And for what I'm showing you right now is allelic richness, where we do have heterozygosity results, but for the purposes of this talk, I'm going to be excluding that, but just know that the findings that we had with heterozygosity was similar. So what we see on the y-axis is the coefficient estimate of the regression, and here we are looking at the significant differences we see in fishes, amphibians, mammals, and reptiles that I've highlighted. And we see that migratory fishes, amphibians, mammals, and reptiles were significantly less genetically diverse than their non-migratory counterparts. And this may be due to a number of things. Maybe due to fragmentation occurring in multiple environments. It also may be due to harsh environmental conditions such as hurricanes, drought, cold fronts that can disrupt migration events. And that tends to be more common in terrestrial biomes compared to aquatic. Another explanation is the differences we see in the incidence of philopatry. For example, in fishes, many fishes return to their birthplace for spawning, and in amphibians, the dispersal of juveniles is the primary mode of population connectivity. And together, these behaviors can limit your allelic exchange and ultimately restrict the pool of your potential mates. However, we saw something different in birds. Migratory birds had shown to be significantly higher genetically diverse than their non-migratory counterparts. And this may be due, again, to a number of things. One being the increased vigility is known to occur in avian species. And vigility is the ability to move from place to place or to disperse, which means that more agile birds can move farther to effectively mix their alleles. And also, possibly birds may be easier for them to explore new habitat in the face of fragmentation events. Another thing, again, another explanation maybe is that the incidence of philopatry. For example, in birds, typically first-year breeders do not return to their hatching site, and natal dispersal is common. Of individuals is also common, and nest site relocations is not uncommon for adult birds. And together, these behaviors can result in a more panmictic population, which in turn means an increase or a larger effective population size, and thus an increase in your genetic diversity. Next, our second objective was to determine if the relationship between biome and genetic diversity differ between migratory and non-migratory species. And we hypothesized that migratory behaviors will influence genetic diversity and the relationship between biome, and such that the relationship between biome and genetic diversity will differ between migratory groups. And our hypothesis was supported by a number of studies that had shown low latitude habitats, or generally warmer habitats, are more climatically stable, and also studies that had supported that geographic areas are positively related to population size. And to do this, to test our hypotheses, we quantified the range of each species in terms of biocomposition. So first, we got species-specific range maps from IUCN, and then we estimated the spatial contribution of terrestrial biomes, each with distinct biota and ecological conditions using the data from World Wildlife Fund. And here's a brief distribution of our data among taxa. And then to evaluate the differences in genetic diversity between migratory and non-migratory, or to evaluate the effect of migratory behavior and biome on genetic diversity, again, we use a linear model. However, in this linear model, we use non-metric multidimensional scaling, which is used to help collapse all the habitat variables into a few vectors, much like PCA. Again, we bootstrap the linear model coefficient estimates to assess significance, and the data, to avoid over-parameterizing our models, data was subdivided by class and run in separate regressions. Okay, and looking at our results, I'm going to be talking about some of the significant relationships we saw in some of our taxa. And let me take a minute to explain what is going on here. So on the y-axis, we have our genetic diversity that is represented by the number of alleles. And then on the x-axis, we had the change in the biome characteristics, which we interpreted from the change in our NMDS axis 1. And we're interpreting that as the change from poles to equator. So looking at this, we can see there's a significant relationship in your non-migratory fishes that we see with the black lines. So non-migratory fishes that are closer to the equator were significantly more genetically diverse than fishes that were closer to the poles. And we see in the migratory fishes, we did not see a relationship. And we think this may be due to a couple of factors, maybe biotic or habitat constraints, but we can't ignore the possibility of an effect of artificial stocking, where stocking can increase the movement of alleles that would not otherwise occur. Okay. And in amphibians, we see a significant difference in the migratory group in the red line, where we see migratory amphibians closer to the equator were more genetically diverse than migratory amphibians closer to the poles, and see little or not much a strong relationship between the non-migratory group. And because we did not detect the same pattern in migratory fishes, birds, or reptiles, we detected no relationship in the less mobile non-migratory amphibians. Perhaps this means that amphibians may be more sensitive to human-caused habitat fragmentation relative to other vertebrates. In migratory mammals, we had significantly less genetic diversity in general in the habitats closer to the equator than habitats closer to the poles. And again, we don't see a relationship in the non-migratory group between poles and equator. And considering that we saw something different in the migratory amphibians, of kind of an opposite opposite relationship, where migratory amphibians were less diverse in habitats closer to the poles, whereas mammals were more diverse in habitats closer to the poles, we suggest that perhaps mammals are better able to utilize corridors to overcome fragmentation that may be present in temperate regions. And therefore, we determined we can't predict relationships between biome and genetic diversity, and between migratory and non-migratory species based on geographic area or climatic stability, and perhaps suggest that other forces may be influencing genetic diversity as well. So to conclude this study and to give you our final conclusions and remind you what we did, we found that the effect of migratory behaviors on genetic diversity vary between vertebrate classes. Again, this may be due to differences in terrestrial or aquatic biomes, the vigility of a species, and or the incidence of philopatry. Again, the biomes impact migratory fishes, amphibians, and mammals differently than our non-migratory habitats, and these may be class-specific responses to habitat changes. And habitat fragmentation and other anthropogenic events may likely be driving changes in effective population size and thus altering genetic diversity. And with all that, I would like to thank our funders. I would like to thank Janna Willoughby for heading up this massive project and everyone else who provided comments. Thank you.